So my name is Sam. I do a bunch of population and evolutionary genomic work, primarily on canids. And truly inspired by canids, my research has no focus. So every time I see a new shiny object, I'm like, my God, this is something I need to get to know. 2 months later, I'm like, something else. Yeah, sure. Everything. Any new. And you will see the through line in a second. This is called magpie syndrome. I do have magpie syndrome. I see a shiny object, I run to it. And like I decided in the morning, this is going to be a 10-minute presentation, but I have 40 slides. So hopefully we'll get through this. And I will turn the motor on by 10 minutes from now. So we have 4 major lines of research in the group. And the primary line of research, where it's going to take up a bunch of the time that I'm going to talk about, is on canids. And then we have this EU project that we coordinate called FindingPheno. So there's some work related to that as well. And for CEH, we have some models of microbiome evolution that we are working on. And finally, there are some straggler projects which can be broadly categorized as population genetics. For Canid evolution genomics, we don't know dog domestication work at all. But I really wanted to put this slide in because the first day that I was, no, first month I was here, we were working with some data from Tom and Mikkel, and the first tree that I made up had the Danish wolf as the root of all dogs, and the Indian wolf as the root of all wolves. I was like, my life's work is done. I should move on. But after that, we stopped working on. We don't really do any dog domestication. I just want to bring the story up. We do a bunch of work on evolutionary relationships between the various canid species, and that early work about 4 or 5 years ago now has led to a bunch of investigations on relationships between the different lineages that are there in canids. And so we focus on a couple of these canids today. But over the past few years, we've been looking at many isolated populations of canids. Uh, the common theme here should be isolated populations, but if you look at the pictures of the specimens, you would think that it's bad taxidermy. But anytime we see a sample that looks like it should looks like this, not very good, we take it up and then we kind of look to see where it falls among the diversity of wolves and dogs. So one of the projects was led by Hasmin and Martha a few years back, where we looked at Sicilian wolves, which are now extinct, and then we looked to see where they fall among the diversity of wolves in Europe. And as you would expect, they look like Italian wolves. They don't look particularly much like mainland European wolves, but we also see here they are, and here are the Italian wolves, but we also see a bunch of dog ancestry in them. So when we look at them a little bit more carefully, towards the end of their stay on this planet, when before their extinction, the last few years, we see that these wolves can best be modeled as a mixture of wolves and some dogs that we have, especially ancient dogs that they shared their range with, so in the Italian peninsula and around the Adriatic. We do similar work on other species as well. I'm not going to dwell too much on it. Again, another excellent case of taxidermy here. So we have some Japanese wolves where results show similar signs of dog-like ancestry in these Japanese wolves as they were getting extirpated from Japan. They are extinct as well. And the nearly extinct Mexican wolves, which also show evidence of ancestry from other related canids around there. Much of this work was led by Jonas for the Japanese wolves and Herman and Hasmin for the Mexican wolves. And today we have a couple of different projects, large-scale projects on canid evolutionary genomics that are underway. One of them is to look at this fascinating creature. It's called the golden jackal, not particularly golden, but more golden than the wolf. And so it has a range which spans all of South Asia, Southeast Asia, and towards West Asia, finally ending in some of Europe around the Balkans. And this range is now, with changes in the dynamics of what species are available now in Europe, especially with the disappearance of the wolf in the last many years, and the heating up of the planet, so that some of these ranges now become accessible to a species like the golden jackal, which is primarily in arid and semi-arid conditions in South Asia, we see that this particular species has expanded its range away from the Balkans all the way up to all the rest of Europe. We don't have too many samples from here, but we do have some samples from Western Europe as well, which are part of our study that Nuno was leading. And when we look at what this looks like, Ignore the fact that this is a PCA, and we are looking here at primary dimensions of variation, and each of the dots here corresponds to the large populations that we've sampled across its range, across the golden jackal range. And we see this axis of variation going from Europe and going into here. This is Israel, so going into Israel. But this particular axis is driven by oversampling in Israel. The real axis is around here. So we see variation going east to west. And when we investigate this further, it looks like a classic model of founding and expansion. So we see that as we go away from India, so on the x-axis here, you have the geographical distance from India. And on the y-axis, you have the genome-wide heterozygosity, so a measure of how large your population is in some sense. And as you go away from India, there's a linear decline of genome-wide heterozygosity. And this is pretty classic observation when you have when you have the center or origin of a particular species, which here is putatively India, and you go away from India and you see that subsequent rounds of founding of different populations, first in Southwest Asia and West Asia, followed by the Levant, and then finally Europe, has led to loss of diversity in every round. And then you see this classical pattern of reduction of diversity as we go along away from the center of a species. So all of this looks great. Now he also looks, Nuno is also leading another study where he's looking at the African golden wolf, which until recently was also classified together with the golden jackal. But in recent years, including a paper from us about a few years ago, proposes this to be a new species, and there is genomic and mitochondrial evidence for much of these findings that this particular species is now a different species called African golden wolf. And when we look at African golden wolf here in purple, we see that they are, as also found in some of the earlier studies, they don't really cluster together in any one spot. And further investigation also shows that they share across the range, they share a wide array of ancestry between the golden jackals, the Ethiopian wolf, and the wolves and dogs. So one of the proposals that we have, that it's a hybrid species, meaning that this is a species that came as a result of coming together of two different species, and followed by reproductive isolation. Although, given the history of canids, where reproductive isolation, even amongst largely separated species, is not very common, this is not such a easily accepted hypothesis yet. So that was jackals and golden jackals, but we are also looking at one of the biggest data sets that we have is put together in collaboration with Tom and Mike Martin and other collaborators in Norway, where we have a sample of around 1,500 wolves from across the range of wolves. We have all of these samples as part of our data set, but you can see that there is quite a bit of oversampling or overrepresentation of samples from Europe. And here our question is now looking at the fine dynamics of population changes, turnovers, and admixture and other population genetics metrics across the range of the wolf. And this work is primarily led by Shin, who recently left us and started a job at the hospital. So here we see that when we look at across Europe, again, some of these results are driven by sampling biases across these different areas, where there are high representation of samples. But clearly there is quite a bit of distinction between the different populations, as you might expect when you look at Europe. So you see distinct populations present in Scandinavia, and the three parts of Scandinavia show distinctness among themselves as well, followed by separation of populations as you move across the range here into West Asia. And this is also recapitulated somewhat by these plots, which are admixture plots, which I'm fairly sure everybody is quite comfortable with and has seen before, where each column represents an individual and different colors represent different ancestry components as estimated from their genetic data. And when we look to see what kind of migration patterns and corridors might exist across Europe based on this large collection of samples across all of Europe, we mostly recapitulate what we see in terms of geographical barriers. You can see, not too much excitement. You can see here that a bunch of these cold zones, which are the blue zones here, are high migration corridors, and the red or brown zones here are barriers to migration. And so you can see that across particular parts of Europe, which are kind of overlapping with geographical barriers, such as the Alps or the corridor here in the Caucasus, and here this is just a refugium, so it's not quite connected as yet. So you can see that some of these results kind of recapitulate what we know from both the geography and movement of wolves across Europe, and they kind of fit very nicely with what we know about modern day wolves in Europe. And when we kind of look at European wolves and try to cluster them separately, we again here in this particular figure on the left, we get back the results that we see in the original picture where we had hit the admixture of PCA results, where geographically similar populations tend to group quite close together. And one of the findings that I think is quite nice is when we look at what kind of ancestry has given rise to the distribution of wolves across Europe, we find that there are two main sources of ancestry when we look at historical or ancient samples as proxies for populations that might have existed at the beginning of that spread of wolves in Europe. We look at two different populations, one of them with a Central Asian lineage coming from somewhere in Kazakhstan or in that particular region of the world, and another one which is got to do with Pleistocene or early Holocene wolves from Europe. And these two particular lineages both contribute to uh, to to the diversity that we see in Europe today, and we see that there's a north-south gradient of these ancestry as we move across Europe. The northern wolves tend to have more of a Central Asian lineage, and this is, we think, primarily driven by the replacement of some of the original populations of north from migrants coming in from Russia. And here, these are the populations in Europe that still show some remnant of Europe-like ancestry from the past. And we finally, in the Canid evolutionary world, there is one more project I think is quite exciting. So this is Lauren's work, and Lauren's primarily completely the driver of both the idea and the work that's done here. Lauren figured out there was this gene called TSHR, which is a receptor for TSH, thyroid stimulating hormone, which plays a thyroid stimulating hormone plays a quite an important role in lots of biological processes. But one of the roles that it plays is ability to distinguish between day lengths in some sense. And so one of the ideas that she had was maybe this is connected to how we see non-seasonality of reproduction in dogs, and in Indian wolves, you see different timing of reproduction compared to different timing of getting pups compared to other Holarctic wolves. And putting these two ideas together, she investigated this gene further. And when you look at this particular gene, you can see that here we have haplotypes of these genes across parts of the TSHR gene. This is the entire gene. And when we zoom in on this particular region where there seems to be quite a difference between the haplotype that's carried by the gray wolves or the haplotype that's carried by the dogs, she also tried to look at what kind of differences do we see when we scan this for different measures of selection. And there is some evidence that this particular region right across on second exon on TSHR gene seems to be selected for some particular process. We can't really put our finger on what is the biological process or the function that it's playing, but our current guess is it's through the pathway. When I say our, it's Lauren's current guess that is through the pathway that's leading to identification of day length and therefore seasonality. Okay, so that was all of the Canid work. I've just gotten a signal that I have 2 minutes, so I'm going to move into my Indian speed again. So then we have another project called FindingPheno, where we're looking at methods for multi-omics integration. This is a collaboration between several different institutions, of which KU is one. The work that's been done here is some way of looking at different multi-omics data sets, primarily with the inclusion of microbiome, and seeing if we can figure out, we can go from genotype to phenotype. So variation in omics to variation in phenotypes. And thanks to HoloFood, we have a bunch of data, which is lovely. Without good data, I can't do any of this. So please pay attention to your data. And then we have a couple of different ideas there. One of them being we use something called causal models, which are basically ability for us to assign causality to the genomic variants that are leading to some phenotypic changes that we see. And in this particular example, this is done in salmon and body weight. And we see this overabundance of hemoglobin and hemoglobin pathway genes that show up when we look at variation that seems to be associated with body weight change. and this is statistically significant. And when we look further, we see that there is evidence that body weight and body size and hemoglobin do share some kind of a very close association to each other. Along with that, we are also looking at graph-based ideas for incorporating either genes or sample-based graphs to look to see if we can identify clusters of genes or clusters of samples, and thus which are more correlated with each other than an average cluster, and thus we can start to make some sense of some ability, thus improving our ability to predict either a phenotype that we care about, like body size and semen, or in the other case where we're looking at gene networks, we want to identify and use uh, information from public databases like KEGG to identify which pathways of genes seem to be primarily responsible for changes in phenotypes. But there are two more things that I want to talk about before I'll cede the floor. Uh, the one bit I want to talk about is microbiome evolution, and here it's primarily Francine who's leading the work here. We are being inspired by work done by Joan Roughgarden, and where we look at, she proposed a model where she was talking about different horizontal and vertical transmission profiles and proliferation of microbiome in a single host, single microbe system. And Francine's been working on incorporating multiple microbes into the system together with microbiome community modeling through one of our colleagues in Finland, where we are looking at different types of microbiome community models and incorporating that into this, and together with host interactions that were immune-based changes that might lead to changes in host and microbiome interactions, and to figure out if we can use the entire system together to say something about changes in neutral changes in microbiome evolution across large evolutionary times. And finally, there is also work done by Barbara on social structure and genetics. So given that IUCN and other organizations are starting to use genomics as a viable alternative to measure the conservation relevance of different populations, uh, the question that Barbara is posing is, does social structure and mating structure changes, do they affect some of these genetic measures? And how should we interpret them in light of that? And therefore, if you look at different primates, different species, they have different mating systems, different social systems. And the question now becomes, do measurements that we compute under the assumption of panmixia and no social structure, are they valid when we look at different species and try to say something about their genetic health? And the quick answer is no. You should be quite careful when you're trying to say anything about species when you have social structure in them. And with that, I think that's the last slide I have. I'm happy to take any questions. Thanks for listening.